First up, we have Professor Tim Brady. We have Dr. Daniel Huang, and you might recognise her from such videos as the one you just saw, celebrity scientist, Associate Professor Jessica Ma. Give her a round of applause, please. Might just get started by, if you could just introduce I will use my microphone here too to make this testing good. Jess, I might get you to start by introducing yourself, tell us a little bit about what you're working on and what your interest is in genetics.
Thank you very much, Joel. Good morning, everybody. It's so great to see you. I'm Jess from the video. And maybe you could tell me what I do if you were paying attention. So I'm from the Australian Institute of Bioengineering and Nanotechnology. I run a group in bioinformatics, which essentially is modeling data from the genome. My work is primarily involved in gene expression, which is the next step on from DNA and mutations. And to really understand why gene expression is important, you can just look to your own body. And we can see that cells in our brain are really different from cells in our heart or our lung. And all those differences are purely due to changes in gene expression. So I'll sign off there and let Daniel introduce himself.
Hi everyone, I'm Daniel Huang. I'm just coming from across the other building. I'm from the Institute of Molecular Bioscience. I work on the genetics of taste perception. So I study a lot of what's the genetic variation within people taste and olfactory receptors, and then use that to understand why your friends love coffee and then you don't, why this guy eats more sugar than the others. They just hate it. And my research is about using the big data and to understand individual differences in their taste perception and how does that relate to our eating behavior and health. And I believe there might be a big continuity to what we are doing today, to do genetic engineering, but I'm thinking of the future is more like what we can do with to apply genetics in personalized nutrition, so we can tailor people's eating behavior so they don't need drugs, but they just eat healthier based on their genetic profile to everyone live healthy. Thank you.
Good morning, everyone. Hi, I'm Tim. I'm here at QBI. I'm a neuroscientist. So my lab is primarily interested in the building blocks of memory. We are super curious about how the brain changes with experience across the lifespan. How does our brain adapt? How does that change with time? How do we encode information in our brain? How do we retrieve it? How does it remain stable from early life all the way until you're really old? So we're super interested in that. We study it in mouse models, preclinical models of learning. Some of the focus areas of the lab we're interested in things like phobia, right, or post-traumatic stress disorder. How does the brain change when people develop these psychiatric disorders? One of the big pushes and why I'm here today is because we're also using genetic engineering to enhance brain function and specifically to enhance memory. Right? So that's one way that we're using this new technology that everyone's starting to appreciate, right? To tune the brain to make it better and function better over time. So I'm really looking forward to hearing your thoughts and talking about some of these interesting issues with you all. Thanks.
Amazing. All right, so you've got a bit of a launching point there. You're talking about genetic engineering to change brain function. So what are some of the places, when we're talking about genetic engineering for humans in particular, what sort of things are you wanting to change? What does this look like?
Well, I mean, in the context of being able to maintain strong memories and perhaps even enhance our capacity to learn new information. That's one way that we're thinking about genetically engineering the brain. But another way that we could think about is in the context of adapting our brains and bodies to new environments. Right? So if you think about Elon Musk and his idea about getting us to Mars and being an interplanetary species, right? Some of the problems that we're faced with is whether or not the human body and brain in particular can survive with deep space exposure, right? So the radiation that occurs from traveling outside of Earth's atmosphere. So there are ideas now that people are studying the possibility of being able to genetically modify our brains and bodies to protect us so that we can survive in deep space and survive on Mars. So that's one way that we can actually use genetic engineering to our advantage, is to adapt our own bodies for changing environments. Any questions? Think about that. I can tell you how we're thinking about doing it. Want to know?
Yes.
Okay, all right. So there's a little creature that you can find in ponds, in puddles, and lakes all over UQ and in Brisbane. They're called water bears. You ever heard of them? Tardigrades, right? And tardigrades live in extreme environments. So there are folks that are actually harnessing the power of the tardigrade by extracting the gene from the tardigrade that protects it from the negative effects of deep space exposure, and introducing that into the brain of mammalian species, right? So that we can now genetically modify a mammalian brain, our brain potentially, with a tardigrade gene, so we now could then possibly be better adapted to survive in extreme environments. That's kind of cool, isn't it? Yeah.
Yeah. Yeah, so from my perspective, I don't know if any of you know your parents ask you to eat spinach, broccoli, those dark green vegetables that you don't like because it just tastes yucky to you. Potentially, we can do something easier, simpler. Instead of telling your mom that, hey, I don't want to eat it, I just do a tiny switch on your gene and then make everything taste slightly different to you. And then, broccoli might taste like, think of a food that you like. Yeah, and then, everything changed and then everyone can eat healthier. And then I think there's something that I envision that genetic engineering can potentially, we might not need to do that. We can modify, we can genetic engineer the vegetables, but we can also do it ourselves. There are different ways of doing that. But this is one way that I can think of that application here, create a better future of health.
Has anyone ever done that? Have they modified the taste profile of a vegetable? So imagine if your broccoli tasted like sorghum.
I don't think the broccoli, but actually yes, I think in the industry they do that. So broccoli and Brussels sprouts in the 90s, they're definitely very big. But what you have now, what you can find in the market right now, they are definitely not as big as before. Maybe you should ask your parents if that's different, but I'll say yes.
It's still gross.
So do you think that's a way to enhance survivability of the species by modifying the foods that we eat?
Well, have you guys heard of the miracle berry?
What's that?
Yeah. So I'm not sure about the chemical compound of it, but it's basically designed to alter your tongue so everything tastes sweet.
So you turn the sourness to sweetness.
Exactly. So I don't know if you've been to a miracle berry party, but you go in and you take this. It's a fruit, right? And so sometimes the fruit's not available, so you can get the tablet version, which doesn't look dodgy at all. But and then you take this and then you graduate's all ready to and then you taste all these fruits or foods that are sour or salty or sweet. So you end up like just drinking vinegar and it tastes really sweet, like juice. And it was originally used and probably is still for cancer patients who don't have very good tolerance for food post chemotherapy, right? But I mean, maybe that's something to put back to my colleagues here. So, miracle berry is one example of miracle fruit is one example of where we're making that modification more locally, right? So taking our tongue. And I love the idea of, water bears in space. I think the water bear on the rocket ship is the next movie coming, right? Snakes on the plane. Is it necessary to modify the brain? Like, could we not just have a shield of water bear skin and be able to withstand space, even though that might not be very attractive?
sounding a bit like a Disney villain there. I just want to point out.
Do you want to say nights?
I think that the idea of adapting our bodies and brains to any environment would be a benefit, right? That's one example, but thinking about enhancing memory. I mean, if we could enhance our capacity to store information through some genetic alteration, would you do it? I probably would.
So if we could give you perfect memory, you pop a pill, we edit your genes, and you've got perfect recall. Tim's got some contraband inside his jacket here. Who would take one of those pills this morning? Who would not take one of those pills? Yeah. Why wouldn't you take it?
Just because like something bad might happen. Say you get into a car accident, and then you're just left with that image constantly. Like your passenger with like a glass shard impaled through their skull.
Oddly specific, but I like it.
It's stuck in your brain for the rest of your life. You wouldn't be able to get rid of it. Whereas if you didn't, there is that chance it just disappears from your mind.
And that might happen, that's true, especially if you use direct genetic engineering, right? Or if you modify DNA. But do you know that since the COVID times and the mRNA vaccines that people have started to appreciate, that's a different layer of what could be considered genetic modification, right? So you could actually use RNA-targeted approaches, which doesn't leave a long-term trace or scar on your genome, but it could still give you temporary gain of function or a more selective gain of function, right? So you could target RNA to go into different compartments of specific cells, and you can even program RNA. We do this in the lab now. We can program an RNA therapy to go into a specific cell and then go into a specific compartment of the cell only when that cell is talking to the other cells. So when the neuron's firing, that's the signal that causes the RNA therapy to go to where it needs to go. Right? So that's super high resolution, but the point of that is that it's not permanent. Right? So you get temporary gain of function, which would be cool, right? And that's all RNA based, not DNA based.
This is a point of order.
Just don't take it while you're driving, then. Just don't take it while you're driving.
Yeah, exactly.
Do people understand the difference between DNA and RNA? Yep, everyone's nodding. Guy, you've all done your pre-reading. Fantastic. Right. Can I just come back to, we're talking about eating healthy by making these healthy foods taste good. Why don't these healthy foods taste good? Like if green leafy vegetables are good for us, and why for some people, they can eat coriander or they can eat these things, and some people can't. Why are we not the same?
So our sensory taste perception, it's a evolutionary consequence. So like people hate bitterness because in the nature, things they taste bitter might be poisonous. So we sort of develop the ability to hate those bitter substances, and then we like sweet food because they are usually high in energy content. That's why we prefer sweetness but not bitterness. And there might be people who hate them more. They might have the gene that make them more sensitive to bitterness, and they could be due to they were in an environment that they just more poisonous food around, or their body are more vulnerable to those substances. So for other people, they are fine. It's not bitter at all because their body can handle those bitter food. So it's a consequence of gene and environment, and that's why we see the individual differences between you and your best friends.
But I would argue sometimes that people actually crave leafy green vegetables when their body sends a signal that they're a bit depleted. All of a sudden, those things start to become attractive again. Right? So if you go on a diet of burgers and fries for a couple of weeks, you probably start to want to have a salad at the end.
You've all come home from school and thought, you know what I want now? A big bowl of kale.
A bowl of leafy greens, right? But the body probably adapts in time, right? So that may not necessarily be a genetic driver, but there's something called epigenetics. Have you guys ever heard of epigenetics, right? So these are changes in the way our genes can turn on and off without a change in the underlying code, but there's a genetic, or it's not a genetic, there's an epigenetic or chemical change to our DNA, which then is really sensitive to environmental changes, right?
Yep.
So an epigenetic switch, like the dimmer switch on a light bulb, can go up and down depending on the environment, right? And so diet and dietary preferences might change in an epigenetic way as well, temporarily.
Yeah.
Right? But there's still, the underlying bias may still have a genetic cause, right? But that could be another way, sort of, just to finish that, that might be another way that you could use genetic engineering. You don't have to target DNA per se to correct the code. You can actually target the epigenetic state of the DNA. So that's, again, another way that it's not necessarily permanent, right? But it could be another way that we could harness genetic engineering.
Yeah, I think that makes perfect sense. Like, people from children to adult, we see the variation. I mean, the gene is the same, but their mRNA, other epigenetic profile change over time. And that's why I can't always make a perfect prediction based on someone's DNA. 80% of the time I'm correct, but it's definitely sometimes I couldn't get it right.
So if we were to remove this, let's say we make everybody leafy green vegetables taste just as good as chocolate, are we, taking something away from humans that, we're more easy to poison? I mean, maybe by that guy up the back with his oddly specific murder scenario.
I think that's because right now we live in an environment that we are safe to, eat leafy green. We are not living in a wild. If we remove our ability to detect weakness and then you just drop yourself somewhere on Google Map, I think there'll be a big problem.
Jess, are there other genes that are sort of both good and bad, or are there things that?
Yeah, I think it's great to mention that because, I think we know a lot about the human genome now, but I think it's worth mentioning that, a genome is not quite plug and play, right? And, my colleagues at Daniel knows a lot about evolution, right? And so it's played with our genetics to get a state, maybe semi-optimal. But, there's examples in human genetics, especially from disease. So you may know that people with sickle cell anemia, horrible disease, but because of the nature of the mutation, these people are resistant to malaria. So in a way, one person's superpower, if you can call not getting infected with malaria, it is actually, with a chronic, horrible disorder. So, and I think genetics is like that. We are robust to a certain condition, but like Daniel said, if all of a sudden all these poisonous, venomous things came up, then we'd be very fragile and vulnerable to that change.
So if let's say I did decide to edit my DNA, and I came to you and said, I want to get rid of my fitness gene. Could I do it? What would it look like? Is it, like a Spider-Man movie? I just take an injection and I get superpowers or turn into a lizard, or what is it? How does it work?
I can give you a bit of insight into that. So genetic engineering is pretty tough, right? So if you're thinking about the human body, and how many cells do we have? 200 billion?
200 cell types, but
cell types, but in terms of across the entire body. So as an adult or a young adult, you could imagine how difficult that would be to target all of those cells to modify the genome of every cell, right? So there's issues with that. But you could, in fact, if you knew exactly what cell population you needed to target, and only modify that one in a very directed way, the brain is our area of interest, so I'll use that as an example, you can zero in on that with genetic engineering, right? So you can tailor the modification to be directed only in a specific cell type and only into a specific region of the brain, for example. Right? So that there's lots of challenges there. How would you get that into the brain? So some ways that folks are using to get genetic engineering technology into the brain is to harness the power of viruses. Did you know that? So you can strip back the HIV virus right? And remove all the bad stuff that makes it self-replicate. So you remove that machinery, and now you have basically what they call a vector that we can then insert our genetic modifying target, or our cargo, which is called CRISPR. You guys have heard of CRISPR, right? So imagine you take the CRISPR cassette that you've engineered with the target that you want to modify, right? It's all based on sequence code. You put that inside an HIV virus that's not going to replicate, right? But it still has the viral properties that allows it to infect cells. So this is how we do it. So we inject either directly or into the bloodstream. Again, these viruses, with a couple other interesting tricks that we can use, can cross the blood-brain barrier and integrate into the cells of interest, right? So you can use viruses as the vector or the system to which you can put your cargo in, and that's how you can introduce genetic engineering into select cell types in the body and brain. Does that make sense? Any question? Yes?
Do you think CRISPR is a viable method for genetic engineering in like a preventative sense for like memory-related conditions that you could get in old age?
Absolutely, absolutely. So CRISPR, there's different versions of CRISPR, right? So there's the CRISPR-Cas9 system, which bacteria use to correct mutations in DNA or prevent viral integrations into DNA. But then there's the CRISPR-Cas13 system. Or then there's even some synthetic versions of it that we work with in the lab that are actually targeting the RNA, right? And we know absolutely for sure that you can use that platform to do exactly what we're saying, preventative measures, rescue memory when it's lost, promote neuroprotection. We can do all that with the RNA targeting system, which is much more amenable to programming. Targeting the DNA is actually a little bit more difficult. But don't get me wrong, there are groups in the US now that are actually creating the technology to cut not just single bases, but like entire insertions and deletions. Massive stretches of DNA are being swapped in and out. It's called prime editing or something like that, but that's one way. Yeah, go ahead, keep going.
Would that have like, how would you mitigate like the impacts that could have with like editing like massive like would it have like a substantial?
Well, if you sorry.
No, I wanted to ask, that's a great question. And is it clear with you and your colleagues what you're actually targeting? Like, it's great to have a system to target, but is there clarity around those targets and whether there won't be off-target effects, or there won't be developing resistance or compensation in the cells that you're targeting?
That we don't know. So we don't know whether or not there's compensatory mechanisms at play. But we do know that it's very selective. So the guides that you use can zero in on a change within a 20 nucleotide span, right? So that's the targeting system that we use. I'm pretty sure that they're actually getting higher resolution by using multiple guides that can then overlap to really zero in on one locus.
So you think, like, as in targeting a specific spot on the DNA strand to change those specific base pairs.
Exactly. So to prevent the off-target effect. So you don't want it to randomly integrate or base pair at some other reasonably complementary sequence in the genome. So the way they do that is through redundancy in the guiding of the system. But again, that's also the reason why targeting RNA is probably a better approach because it can avoid those long-term catastrophic effects on the baseline DNA code.
Yeah, I guess, like, from a research perspective, we're still understanding, what genes do and how their expression codes for these really complex behaviors, right? Like memory retention or phobia. And while it's really cool to have that precision of tools and be able to work with like animals and focus on the exact brain cell, I think we also recognize that response is whole body, right? And so there's coordination, maybe it's a cascade, but there's still coordination between, even your adrenal glands and your skin. And so I think, making those changes up there in the hierarchy and seeing how, as a species we adapt to that change would be a really interesting kind of question.
So maybe it's better to adapt the environment to us rather then adapt us to the environment. What do you guys think? Just modify the environment to make it more hospitable so we survive, or would it be better to tune us to the top corner and then we'll come back to the middle? Yeah.
On that note, what's your stance on using like Cas9 or Cas13 on animals or parasites or something like that?
To prevent like malaria sort of thing.
Yeah.
I mean, I don't have a problem personally with the idea of gene drives to render the mosquito that carries malaria incapable of spreading it. I think that that's totally fine. I don't know what that's going to, I can't predict what that would be in 10 generations in terms of how the ecosystem will respond to removing that from, what is the benefit of transmitting malaria? Is there a benefit to the ecosystem? I guess the question is, there must be some reason it's there because it survived evolution.
What about stuff like what they're doing with oxytech, where they're trying to control mosquito populations, trying to cut down their numbers by making it so they can't mate anymore?
Yeah. I buy it. I would support that in a big way. Right?
Would it be fair to say that I think on the one hand we've got these tools that are getting more and more powerful. On the other, and we've got the ability perhaps to deliberately wipe out a species as opposed to casually wiping out a species like we've done for the last few 100,000 years. But also we're perhaps still catching up on the, you talk about both within our own DNA, like what is all the ramifications, but also on the broader, are all these complementary knowledges catching up?
Can I give you another example of perhaps a negative aspect of gene editing technology? Do you remember a few years ago the CRISPR baby scandal that happened in China? Do you remember that? So they modified a specific gene in these two babies, embryos. They're growing up now, right? The target gene that they went after was the gene that promotes the transmission of HIV. Okay, and because one of the parents was HIV positive, right? So they modified the genes in these babies so that they wouldn't transmit HIV or integrate HIV. Now the problem with that is that the exact same gene that they targeted is super important for a process called memory linking. Being able to have the associations of your memories in time. So in your brain, the exact same gene that's involved in the transmission of HIV is critically important for cognitive function. So there could be a catastrophic effect on these individuals that sure they're buffered against HIV, but now the side effect or the negative consequences that they may have impaired memory abilities. So this is something we have to consider. So just the young lady in the front with the white shirt, you wanted to ask a question.
No, I was just commenting on your question of whether we should modify the environment to us or vice versa.
Yeah.
I just wanted to bring up the point that if we do modify the environment, there's a very fragile balance in the environment, obviously with other species, other ecosystems around us.
Mm-hmm.
So it wouldn't just be impacting ourselves, but everything around us. How would we mitigate these effects?
Yeah, that's a good question.
Yeah, has everyone seen Lilo and Stitch? The mosquito population is very important, right? But I think that's a good point, and there's currently, or has been, field trials being done on dengue, right, where they genetically modify the mosquitoes to make sure they don't transmit that. And I'm not sure what the success of those are or the impact on other species locally and more widespread. I wanted to provide another example, if that's okay, about how little or just how far we've got to go. There's an example in the US recently about cystic fibrosis. Everyone know about this disease? Yeah, so it's single gene disorder, right? There's these diseases called Mendelian diseases that you all know about, where it's one single gene that's gone wrong, right, from birth. And so because of this one single gene, the CF patients, have lifelong chronic illnesses. And so in the US, they have a system where they test babies, right, to see if they're at risk for CF. But what they found was that with the testing, it was inadequately picking up the the CF gene in predominantly African American patients. And that's because the reference that they were using was really based on a Caucasian reference, and also because of the variability in the testing quality across states. Some of them are only testing a couple of loci, others were doing much more comprehensive testing, and the more comprehensive testing was the one that picked up on, well, this kid is black and it has CF, right? And so you can imagine when you're cutting the health budget, what are you going to cut? And so you have these really horrible examples of moms, because this is a inherited disease, right? You had moms who had like 4 or 5 babies who had CF. So when the 5th baby came along, they knew that this child had CF, but they were fighting doctors, they were fighting hospitals, they were fighting insurance providers to basically get their kids tested properly. And so you can imagine in this day and age, right, that America is dealing with this problem. We won't go there. But, I think that is a really illuminating example of what we know in human genetics and all of Tim's amazing, technologies, and yet we're still faced with this kind of gap in access to reaping the benefits of that understanding about human genomics.
And sorry.
Oh, no, I just want to comment on all the fantastic tool technology we have today. It's actually built up on a lot of previous work. Like, if we want to target a certain gene, how do we find the gene? Like, it's actually involved a lot. It could be a work from the 10 lab over the world, and then we discover this one gene, and it could only be related to the European institute, not apply to other institute. And then, so when I was an undergraduate student, I was told that 1 in 10,000 discoveries that would actually work, would be applied to our society. And once we discover the gene, and then we test this potential application, and then it needs to go through very stringent, clinical trial from cell, animals, and then humans. It takes a very long time to ensure that it has minimum negative influence on us or on the environment. So everything we do is to ensure that it have a bigger benefits than the damage it could cause.
Do you have thoughts on, perhaps social access to these type of, if we're talking, let's say we're talking about babies, treatment for babies. You're saying it's different in every state in the US in terms of even just the testing. If we start to think about actually applying gene therapy to embryos, is that going, a pharmaceutical company is going to make that available to everyone? Is that?
Yeah, I remember you were saying that, like, basically, if it's a pharmaceutical company, they need to make money. So only if there are enough customers who can afford to use this technology, because it's not cheap. Because I know the FDA approved CRISPR to treat sickle cell. I think it costs $2 million US. So it's very unlikely that they'll have people. There must be someone who can afford it, but not everyone. And
so do you think they'll draw the line? Do you think they'll draw the line as to where and when one can use genetic engineering? So, to correct a disorder is one thing, or to get gain of function, or to change eye color is another, right?
Who would be in favour here if you had an embryo that was tested and had cystic fibrosis or some other, and it was a simple gene therapy that had been tested and was, who thinks it would be okay or ethical to apply that therapy? Okay. What if it was something still kind of bad but not critical, like being short-sighted, real pain in the arse, right? I've had to wear glasses for a long time. If we had a gene, if we'd be able to test, they've got the gene for short-sightedness, we could change that. Who would be supportive of doing that survey? What about if it was something not too bad, like, I don't know, red hair or something terrible?
That's horrible.
No, just kidding. But yeah, what if we're talking about something cosmetic? If you wanted your baby to have red hair or to have really nice skin, like who thinks it would be okay to do gene therapy? Change that. Fewer people.
So what about the access issue there, right? So there's a question of who can afford this stuff, and at what point do you make a choice about what's allowed or who's eligible, right?
I mean, like, this sounds really bad, but if it really is as expensive as it is, and people are able to afford it, then, like, regardless of whatever the issue is, if they can pay for it, then...
Yeah, I hear you, because it's like a two-tier system, right? So why not? If people can afford it and there's access to it and it takes the pressure off a public system, then why not? It's like private healthcare.
Yeah, and especially if, genetic engineering is getting so much better and so much more common, then hypothetically it should be getting cheaper too.
Yeah.
So like, stuff like disorders, they should be able to be treated for low prices.
But what about the people that use it for gain of function to confer advantage on the kids? Right? So, I don't know how many of you do Saturday maths training just to get a competitive edge over your colleagues, right? But that's a thing. People do that. So what if you can have genetic engineering that allows you kids to be superstars in math or without having the extra tutoring sessions? Is it fair?
Oh, sorry.
Yeah.
So I was just thinking that it would deepen the social divide between say different classes or different economic groups. So that would be a major issue in society if one group could access these and they were richer, and then another group couldn't, and that just bring apart these different groups.
Yeah.
Question up front.
I'd just like to continue on that point. I feel like especially with a technology like this, it's likely that it will get more expensive, like insulin, because they want to drive up the price for even something essential like curing disorders, because we do a greedy capitalist society. And
Particularly if it becomes that the only way you can compete in maths or sport is if you've had.
it becomes more of a strange eugenics issue that only the rich can alter themselves to what they actually want, and it also feeds into the ableism issue. Because ableism is already disability is already associated with poverty and lower socioeconomic status, and it just puts more people at a disadvantage because of the intrinsically just unequal access that people can get.
So I guess the question is, if there is that possibility, then do we just stop what we're doing and not investigate this potential?
But I think the other side of that is exploring this beautiful tension that you picked up on, where ableism, genetic engineering could be a great tool for promoting ableism, bringing everybody up to that level. But then, in the wrong hands, it can exacerbate the disparity between the able and the not able. And so, where does that balance of power hold, or who has the power to shape that, right?
I want to ask you guys a question. Have any of you done genotyping of yourself? It cost thousands, it's $100,000 long time ago, and now it only costs $50 to genotype yourself. And if the government, if the society would believe that having that information can produce more benefits to everyone, then maybe it's not individual who is rich enough can afford it. It should be government when we should implement that in the health system, so everyone can have access to that. If they can actually save the government money from treating cardiovascular disease or something else. And it's not a bad idea to genotype yourself. I know there are a few companies doing that, and they send you a nice interesting report.
Now, we've got about 10 minutes left here. So we're very much going to throw it open to the audience now. Either you can sort of continue to discuss this ethical question, or if you have other questions you would like to keep the panel with, ethics or otherwise. We're open to questions or comments from the crowd. So I saw some hands in the middle, but they've gone down. Yep, we're going again, yep.
Different countries will inherently have different legislation surrounding this, and I don't think we can trust any human society to have this power.
I think that's a fair point, yeah. So I think another way to think about it would be what systems can we put in place to ensure that either trust is there or that we mitigate the risks, right? Is it that we have to become more transparent, right? Or is it that the oversight is much more comprehensive? Or because you're right, movies tell us that the evil science doing this is going to create that underground lab and do all this, but how do we as a society ensure that we create systems where we get the best benefit out of the science that we know?
It has to happen, but I mean, at the end of the day, we've already exceeded the rate of human evolution. 150 years of industrialization. Look where we've come. We've gone from figuring out how to fly to now we're on our way to Mars. Right? So this is the acceleration in terms of our technical advances has outpaced worrying about the long-term path of evolution, right? We're now at the time of our existence where we're going to integrate into, we're going to become, what do they call it, cyber, when you're integrated with machines, right?
Interestingly, last year's session was on AI. We had a very similar discussion around regulation control. Yeah. And if we don't do it, will somebody else do it? And then, how do you handle that?
So we need to, I think we need to actually embrace it and understand it, right? But what is the limit? How far can we go with genetic engineering? We have to go into it so we have eyes wide open to know what we can possibly do. And of course, then regulate, yeah, for sure.
I love the discussion, but I want to sort of hear from a few more people. So I'm going to jump up the back there to that older student there.
I was just wondering whether we can use this to cure the allergies which happen.
Allergies.
Allergies.
Yeah, I think, there people are using, so the mRNA therapies have now evolved to an integration of antibody therapy. Right? So once you identify an antibody that will respond positively to whatever allergen, you can program the system to generate that antibody through an mRNA therapeutic. So it's happening now, absolutely. It goes from that sort of response to infectious disease all the way through to Alzheimer's disease. So there's a group here at QBI that actually has an antibody-based system that recognizes dysregulated tau, a protein that is broken in Alzheimer's disease, and they're administering it as an mRNA therapy. So the mRNA introduced into the body, it starts to spit out the antibody that's required to neutralize the bad tau. Very cool, right? So I think for allergies, that'll be a thing of the past.
All right, questions?
On the ethical thing, because let's be nihilists again. We've been talking a lot about using it for health and science and mad scientists out there misusing it. What about just straight up the governments? What's stopping US, China, or Russia from making a set of super soldiers for their military or something? That's objectively a negative use that they can legally do, and it's very viable to give them the technology to do it if we continue with upgrading genetic engineering to oblivion.
So how do you stop it?
We can't stop Russia from invading their neighbours.
Right? So I think it's better to understand it at its deepest levels, right?
So to add some ways to that, I think the answer is nothing. There's nothing to stop them. But actually, the NIH, the National Institutes of Health in the US, but also here in Australia and other governments, invest heavily in studying malaria because apparently there's an incredible quote where more soldiers die from malaria than bullets. And that's not to say that they're secretly creating a supersonic breed of humans, but I think there is a lot of investment in biological research. And who knows, I'm not trying to add to the conspiracy theories, but I think when you think in that way, there is a bit of a bloodline, right?
I mean, and genetic engineering is just this generation's tool, right? So in World War I and II, the Germans were using methamphetamine and drugs to create super soldiers, right? And in the Vietnam War, they were using marijuana and other things to help kind of buffer the effects of the war. So we're just now in the generation where we have these new tools that are disposable, which are a heck of a lot more potent, obviously. But
I mean, like,
we're not going to stop it.
looking at it from that standpoint, like, obviously you're taking something that was meant for good and then using it for something awful. Like, using the World War II example, like, nobody wanted Oppenheimer to take Curie's atomic level and make it a bomb, but it did it, so, like, nothing stopped it.
That's right. So again, it comes back to being able to understand this stuff at the deepest level.
So back on that.
So more on the technicality, but is there any ethical consideration like doing genetic engineering with plants?
It's regulated, absolutely. Yeah, for sure. There's regulations in place already for that.
And I think so the physical nature too, so there's strict regulations around where you're planting and they like the local environment, I think, and what has access to it.
My partner is a plant breeder, and one of the things to point out is that a lot of plant breeding right now is you take 1,000 of these plants, you mash them together in sort of romantic times, and you create a whole lot more plants. All of that random selection, there's no guarantee that any one of those new plants is safe. In some ways, genetic engineering can actually be more controlled. You can make the specific changes that you're trying to get to. So there is this question of, is some of this stuff safer and easier?
More efficient and less wasteful. I mean, if you think about plants are one thing, but then if you think about livestock, right? Like there's a lot of resources and impact on the environment that comes from breeding livestock, creating new strains, figuring out which one's going to work, and essentially the same, not the same randomness, but there's still, compared to actually precisely modifying aspects of the genome.
And just in terms of the plant thing, just one last little extension. So there's a group in Western Australia that are working on this. There's a Centre of Excellence for Plants in Space. And they're actually reprogramming plants to generate medicines that are good for us. So they're genetically reprogramming a plant to grow antibiotic. How cool is that, right? So, obviously that needs to be regulated, but that's one example of harnessing the power of plants for our benefit in maybe when we're living on Mars.
We've got time for a couple more questions or comments. Gentlemen there.
So, when your baby is an infant in the womb, if you discover that you will have a disease, and you use genetic engineering to alter the disease, does that raise the question as to whether that is ableist in not accepting the child if it has the disease?
That's a good question. I personally, I would probably go for the genetic engineering.
I think that's a really great question.
Control over your environment, right?
I'm left-handed, and you can argue that that makes me very clumsy in a lot of things. I don't know. I think our concept of ableism is also deepening and growing and maturing. And then meanwhile, what's really fascinating is simultaneously our understanding of genetic defects and the consequences is also deepening, and then our options for therapeutics. So I think it's a really interesting time to be, entering science, because you've got the confluence of these three things, right? The genetics, the therapeutics, but also I think our concept of ethics and genetic ethics has gone a long way, right? So I don't have an answer, I just think you really interesting questions to tackle.
Yeah, and there are different stages, like you start from screening, and then what's the time point? Like you can know the very early stage, and then you can make a decision early and really want to move on. And then if you decide to go with that, and then maybe it's a time to consider whether you want to do genetic, treatment through a different approach. And yeah, I don't have an answer to that as well. I became a father 3 years ago, and my partner, we did a genetic screening, and it's knowing your own gene and your children's genes actually quite. There's a nervous moment, like we don't know what will happen, but.
I mean, they've been doing screening for Down syndrome for 40 years. It's pretty common. So we're just at the next iteration of the choices of genetic disorders that we can screen for.
I don't know what the current recommendation is, but they at least let you know that you can make.
Yeah. But I mean, also, I think our understanding of different disorders is also growing. Like my friend works as a pediatric clinical geneticist, and he was talking about, a kid who was really little and diagnosed with autism, and his mom was in absolute tears, but he was saying, this is the same little boy that, before you got that diagnosis, and I think the resources out there, the communities, we're understanding that ableism is not one thing, right? It's actually a whole spectrum or distribution. And
I think genetic screening for autism is super dangerous, though.
Yes,
Right?
that's
Because
all
autism in many cases can be a superpower.
Exactly, and we
Right?
appreciate that.
Should be, yeah, right.
Yeah.
So, but there's no, I don't think there's any. I mean, maybe that's just fine bias, but a reasonable way to, label somebody as being dysfunctional based on a genetic association with autism.
But that speaks to where we've come, right? Because 10 years ago, we may not be using that language, right?
So we're almost at the end. I think that's just, maybe some summing up from the panel. We've been talking here about a broad range of ethical and technological questions. Coming back to our original theme, was the survival of our species. One way or another, do you see that genetic engineering is going to be part of our, whether it's our cells or other organisms? Do you think this is going to be a key part, or do you think there are other pathways?
I think it's essential for our own evolution and our own ability to adapt to the world as it changes. I think we have to embrace it.
I think it's good to have all the technology ready when something unexpected happens. COVID, but we have the technology ready. We have more options to deal with the danger.
I'm going to skip the question a bit by saying that I think we have the technology is growing, it's always going to be here. But what I think in the future is going to happen is we're going to get better about collecting more comprehensive data. And so maybe that starts with you, in that we're capturing individual genes, we're understanding their variation much more comprehensively than having, a billion deeply shallowly sequenced genomes. But instead we use that knowledge, and I think understanding the data better, understanding our own variations better, will have positive impacts into that genetic engineering.
And I think we really highlighted today that there are huge ethical considerations, some small things, some big things, and science cannot tell you what you should do, but it can tell you the consequences of doing a thing or not doing a thing. And so I think kind of having really good data, having really good information available, so that when you all, in a few years, have to vote and make decisions or decide legal cases or consult parents who are coming at you with these difficult questions, should I apply this therapy? Or you be in that position yourself. We need to have that information, and so we're really excited to be working on it here at UQ, and we're really excited for everything that you're going to share with us after this, and all the work that you're going to do, and all the thinking you're going to do on this. So we, of course, will be back to have these next sessions, but we are going to take a break now and have some food. So could you please join me in thanking our fantastic panel for their time? We're going to head outside. How are we going to coordinate that?
Just on the terrace, half an hour. Just
half an hour, you're sitting around?
Yeah.
So panel will be around, so come and have some follow-up discussions. But head outside now, follow your teachers, head out the door there. We'll see you soon. Good.
Introducing Steve the Bee. What do bees like Steve have to offer for us?
Bees are essential for our environment, acting as pollinators for more than 250,000 flowering plant species. Tiny insects like him allow plants, including countless food crops, to reproduce, ensure food security for our growing population, and preserve ecosystem health. Honeybee pollination adds more than 200 billion in economic value to agriculture annually. But bees like Steve and his friends are facing serious threats to their survival, one of the reasons being pesticides. Look at what just happened to Steve's best friend.
He died.
Neonicotinoids are one of the most toxic systemic pesticides in agriculture to our honeybee friends. They contaminate all surrounding groundwater and soil and are easily spread, causing colony collapse. Neonicotinoids disrupt bee nervous systems by binding permanently onto nerve cells, overstimulating them, causing paralysis and death. Recent statistics show that the honeybee population is decreasing at an alarming rate. Here we can see that the US honeybee population has nearly halved in recent years. In Brazil, exposure to neonicotinoid pesticides killed more than 500 million bees in less than half a year. This is putting significant stress on beekeepers to maintain their numbers.
What happens if we don't stop this? What if all the bees die? Countless plant species will begin disappearing one by one, and we risk losing all plant species that bees pollinate. This will lead to agricultural collapse, leading to food crisis and famine, extreme struggle to sustain the growing human population, and death. Eventually, other major pollinators such as butterflies and moths may also go extinct. What if our only option was to have mosquitoes replace these vital pollinators? Would you rather Dave the mosquito replace Steve?
Do you enjoy living as we know it? Yeah. We wouldn't be able to without bees, because what awaits without bees is severe systematic collapse. So, how can we save Steve the bee?
Our hypothesis is that genetic modification to the bee's natural gut bacteria to increase the expression of P450 genes can help bees develop resistance to and detoxify neonicotinoid pesticides. So P450s are a family of oxidizing enzymes. Our P450s are naturally responsible for detoxification in insects, so they can help insects develop resistance to pesticides. If we insert multiple copies of this P450 gene into a bacteria, as the bacteria reproduces, so will this gene, increasing the production of P450 enzymes. The more the bees can detoxify, the less they will be affected by neonicotinoids. Detoxification occurs in a multi-step process where first the P450 oxidizes the neonicotinoid, altering its chemical structure so it can no longer affect the bee's nervous system. Then the bee will naturally excrete it.
We plan to insert the amplified gene through bacterial transformation into a stable plasmid. A plasmid is a small circular piece of DNA, and the plasmid stability will decrease the chance that the bacteria will reject it due to selective pressure. So we plan to host the host bacteria in Lactobacillus sperm 5 so the bee's guts doesn't reject it. Lactobacillus sperm 5 is one of the most abundant bacterium found in bees' gut. We plan to introduce the bacteria through bees' diet, and such as nectar, and to work with beekeepers to ensure that the resistance isn't spread onto pest insects. And to make sure the P450 is there consistently, we will introduce the bacteria to the bees routinely.
Our solution is non-invasive and easily spread through food sources. But most importantly, plasmids are not permanent and easily removable, meaning that in the case of dangerous mutations or dangers to the ecosystem, we could remove it without having to directly edit the bee's entire genome. Furthermore, our solution can also be applied to other endangered pollinators, such as butterflies. However, plasmids could be spread through horizontal gene transfer, meaning there is the possibility of it passing on to pest insects. Furthermore, increasing the P450 in bees may have unforeseen consequences for their bodily health. But overall, we believe our solution has potential.
So, guys, shall we witness the downfall of bees and humanity, or are we going to take action to save our pollinator friends like Steve?
Genetic engineering is a new and exciting field that can help us maintain our global ecosystem, but only if we let it. It can not only help us reshape humanity, reshape our agricultural system, and save our honeybee friends, but help us save humanity as a whole. Thank you.
Fantastic. I think I'm going to throw, is it Jess's?
Okay.
Okay, some questions from our full judges.
Yeah, so thank you for speaking up for Steve. Yeah, thank you. So no, I thought that was an amazing presentation and really great way to drill down to a very, very important problem. Can I ask you a bit more about, I love your strategy for genetic modification. Had you thought about targeting other parts of the bee, not just the gut?
That's a great question, thank you. First of all, we decided to target the bee's gut bacteria because we found that the gut bacteria actually helps the bee regulate their P450 count, so it's naturally already part of the process. So we thought that we could add a plasmid to it and help edit it. It would be more natural to the bee's whole bodily balance as a whole, instead of editing a completely new part.
Great. And do you have any knowledge of what people have already tried in honeybees?
Yes, there has been experimentation with P450s in that, since they have already been linked with detoxification, there's been a lot of attempts to amplify it. We've seen one of the studies that amplified a select part of it to a certain degree. Our plan is to amplify that even more to increase the effectiveness of it and just build on previous research.
So if you believe that P45, having more copy of P45 can help Steve to survive from not same consequences as his best friend, how are you going to design a study or project to test that your approach does work? How do you know having more copy of P45 does increase the survival of the bee?
We could try to track the activity of P450s, or like in general the quantity that's being produced in different bees. So we could experiment with a control group with no edited genes and then some that are edited and see their different responses to neonicotinoids and see if actually editing the plasmid and putting it in increases the quantity of P450, or if the P450 actually has activity and does do more detoxification.
So just a compliment, and that's a fantastic consideration about the possibility of horizontal gene transfer. It's very cool to acknowledge the limitations. With regard to P450, do you know what the side effects might be if you overexpress this thing in the bee? It's not selective for nicotinamides, right? So what is P450 overexpression going to do beyond just enhance detoxification of that pesticide?
It's an oxidizing enzyme, I believe. Yeah. So it might oxidize the other things that they're not meant to. So the bee's natural gut system, it might mess it up or disrupt their natural processes. So
is there a way that you could provide balance? Is there a way to titrate the effect, I guess is the question, so you can avoid the negative side effect profile?
We are trying to address that in our solution, but the problem is that we haven't actually done the experiment, and we wouldn't really know how this would actually play out in a real life situation, in that we're actually editing it in a lab. So in a more comprehensive version of our presentation, we will probably have different combinations and different amplifications to see which amplification has the least negative side effects while still having effective detoxification of pesticides.
There is also a possibility that if the P450 is in excess, the bee might just get rid of it itself and just remove it because it doesn't need it anymore.
Our society faces a critical imbalance between carbon emissions and the natural recycling of carbon. Over the past 200 years, our carbon emissions have skyrocketed, surpassing the capacity of the natural cycle. This surge, largely propelled by our factories, industries, and cars, pumps an astonishing 37 billion metric tons of carbon dioxide into the atmosphere annually. This results in many detriments to biodiversity and human well-being through pollution, the scarcity of vital resources, and destruction of our living environment. Plant carbon sequestration is a large part of the natural carbon recycling process, which stores atmospheric carbon in biomass from our soil. This process helps mitigate climate change, transferring greenhouse gases from the atmosphere into long-term sinks. In this process, plants absorb carbon and convert it to glucose through the Calvin cycle during C3 photosynthesis. A key component of this cycle is the Rubisco enzyme, which catalyzes the reaction between CO2 and RuBP, an organic compound, to create PGA, an intermediate for glucose production. However, photosynthesis is a relatively inefficient process. A key issue with this enzyme is that it bonds not only to CO2, but also to O2 at higher temperatures and concentrations. This leads to an alternative pathway called photorespiration, which creates waste products that actively lose 3 molecules of fixed CO2 when creating glucose. This reduces the efficiency of carbon fixation and is a waste of energy, squandering 25% of the plant's fixed carbon. It also reduces the concentration of carbon the plant can absorb in one cycle, leading to lower rates of photosynthesis. However, if we wish to mitigate the effects of climate change, a viable natural method is to boost plants' abilities to sequester carbon. To do this, we can employ a plethora of genetic engineering methods to boost the ability of plants to efficiently fixate carbon during photosynthesis.
How can we use genetic modification to tackle a problem threatening our species? We propose genetic modification to improve plants' abilities to complete photosynthesis and absorb carbon. To do this, we will increase the enzyme affinity of CO2 over O2, minimizing photorespiration as a reactant pathway, also absorbing carbon more efficiently.
Okay, so this is the active site of the Rubisco enzyme. So we have identified a few major amino acids that are significant in stabilizing CO2 during the transition period in the enzyme. So these are threonine 182, glutamine 409, lysine 210, serine 387, and glutamic acid 213. We have identified glutamine 409 as a very significant amino acid in this process. So it has a carbonyl group and an amide group. And so it exhibits van der Waals forces with the oxygen in CO2. Okay, so we propose changing glutamine to glutamic acid at position 409. We coined this Q409E. Okay, so how is this different? Okay, so glutamine and glutamic acid are very similar amino acids. So glutamic acid has an oxide instead of an amide group. And this means it has a negatively charged side chain, as we can see here. So how will this increase CO2 affinity? Well, we can see that carbon actually has a partial positive charge due to its lower electronegativity than oxygen. And so when we change the glutamine to glutamic acid, we give a negative charge side chain. And so we think that this will exhibit electrostatic interactions with the carbon in CO2. And therefore, this will increase electrostatic interactions and improve the overall stability of the active site. Therefore, this will increase CO2 affinity.
All right, so we hypothesized that by changing glutamine to glutamic acid at position 409, it will increase the affinity for CO2 because of this increase in electrostatic interactions. And our proposal is that we will modify the chloroplast through chloroplast transformation, and through site-directed mutagenesis, we will be able to introduce the vector, which will have the modified base and other homologous sequences, so that will be successfully integrated into the plant cell through homologous recombination. This will be done through ballistic particle bombardment, where we will accelerate the modified vectors using high pressure gas into the chloroplast cells, and it will successfully be integrated using homologous recombination. The modified plantlets, plant cells, will be grown alongside natural tobacco, and we will measure this through gas exchange cuvettes, and the intended result is that we will have minimized the O2 pathway, and that means the genetic modification is successful.
If the Rubisco engineering is successful, a similar process can be applied to a broader range of C3 commercial genetically modified plants such as soybean and cotton. This will be measured by the change in the concentration in the soil and carbon levels. Some challenges we may face: compromising the enzyme stability, which would also affect the enzyme's catalytic ability. This would overall distort the quaternary structure, which means different interactions with different molecules. In summary, the potential benefits of this modification improve the active site for CO2 over O2, increasing the rate of photosynthesis and overall mitigating the greenhouse effect. Thank you.
Amazing. Let me pass you to Dan on the first instance.
I think it's a really interesting idea. Reminds me of my undergraduate project. It's really nice to see you use, I don't know, to use it's probably not the AI to show the structure, but it's really nice how the increase the mutation or the genetic modification can increase the affinity potential of this affinity to bind CO2. My question is, how do you know, how are you going to test how efficient it's going to be? You're saying it's I think it's like two CO2 with 3 was lost in a cycle, and then you do the modification would potentially increase the affinity. How do you test, how do you assess, how do you evaluate this genetic modification would bring back of those lost CO2?
So through the gas exchange cuvettes on the modified plantlets and the non-modified plantlets, we expect to see increased absorption from CO2 because the cuvettes measure CO2 and O2 levels in a sealed-like surrounding of the leaf. And if we see a decrease in O2 absorption and an increase in CO2 absorption, then we can recognize that the modification is successful or not.
Do you guys have any ideas about maybe other plants that would be more efficient carbon sink mechanisms? I'm thinking like longer lasting trees or palms, because soy and cotton and tobacco, it's like a high turnover rate, right? It's seasonal.
Yeah. So firstly, we know that CD4 plants actually have a more efficient, like a Rubisco system where it actually fixates carbon more efficiently. But we think that because C3 plants they're more abundant, and so we think that using commercially significant plants like soybean or cotton, like C3 plants, we think that they will probably have a more global impact, and especially as in we've identified some of these plants as plants that have a pretty manageable genomic structure. They're easy to modify. So we think that they will probably be more inducing for our project in the beginning phase. But then as we continue, we'll have a better understanding. We can expand to a wider range of plants.
Just a quick question then. You mentioned how there might be some issues with messing with that specific amino acid. Do you know if people have tried that before, maybe in other plants, not tobacco?
So this experiment has been tested before, not with the exact position of glutamine and to glutamic acid. It's been tested with valine and leucine. However, it's been not as successful as it should have been because the negative charge that the glutamic acid provides is more favorable for carbon dioxide absorption. So this theoretical plan should be better. So yeah.
I would like to add, so we have seen like scientific literature which has had these kind of experiments done, but we haven't seen any with the exact proposition that we are proposing. So we have gone through pretty deep analysis of the active site and the types of amino acids inside it, and we have decided that this would pose like a modification that would be more favorable to our goal.
So for the last question, so assume this work, and then the plants take more CO2 from our environment, but it basically means the air will be different from what we have now. What could be the potential influence on our environment?
So one way to think about it is this genetic modification of carbon absorption not only reduces the amount of carbon in the air, but it also it sequesters the carbon in the oceans. It removes the stress from the carbon sequestration in the oceans, meaning more ecosystems could live in the oceans. It's more beneficial for the marine life. But in terms of the general atmosphere, reducing the amount of carbon would also mean in soil sequestration, the cycle is just accelerated, meaning more fossil fuels can be used, it's just generally more carbon in the soil as well. So another way of thinking about it would be just speeding up the process rather than removing carbon from the atmosphere and adding more oxygen overall.
There's another way to put it. So it's kind of like a buffer towards the accelerating emission of greenhouse gases. So we think that this could pose a buffer because the emission of greenhouse gases, we think, well, obviously there has been actions taken to decrease it, but we think that some industries, they will still continue to emit even if it's gradually decreasing. So we want to provide a suitable buffer towards the increasing emissions of such carbon.
Atmospheric carbon dioxide levels have been increasing dramatically over the past 50 years. Since 1974, the increase in atmospheric carbon dioxide parts per million has been almost threefold. But what does this mean?
Carbon is the essential backbone of life. It exists in all systems on Earth. It is crucial that it maintains a balance. Why? Well, CO2 is a greenhouse gas, so it insulates the Earth and traps heat.
However, this balance has been disrupted. Carbon dioxide emissions are increasing due to deforestation, burning of fossil fuels, factory and vehicles.
The world is warming, and with more CO2, the problem is getting worse. Global temperatures have risen by 1.2 degrees Celsius since the 1850s. The UN Environment Programme predicts an increase of 2.9 degrees Celsius by 2100.
More CO2 in the atmosphere also means more CO2 diffusing into the ocean, causing ocean acidification. Forests can absorb and retain CO2 emissions, but for oceans, the CO2 reacts with water to form carbonic acid, which lowers ocean pH, dissolving marine animals' shell and exoskeletons, and pushing marine species to extinction.
Current solutions are not working fast enough. Despite significant effort in recent times, levels of CO2 are still increasing. Why? Existing ideas face challenges with sustainability, funding, legislation, but the main issue is effectiveness. Take rechargeable lithium batteries as an example. They sound great, except for each ton of lithium, 15 tons of CO2 is produced.
Current solutions are nowhere near enough or fast enough to limit warming, let alone reverse it.
But there is a solution.
Jean, Kiadi, and Erica, and our solution harnesses nature to capture more carbon dioxide.
Okay, so we hypothesized that the enzyme ribosco in eucalyptus trees can be genetically modified, which can drastically improve the trees' carbon absorption, which thus reduces global warming.
The ribulose biphosphate carboxylase, or ribosco enzyme, is found everywhere. Located in chloroplasts, these enzymes are vital in the process of photosynthesis, as shown by the formula.
However, the enzyme ribosco mistakes oxygen for CO2 around 20 to 40% of the time, and wasting over 30% of photosynthetic energy.
The arrow here points to the rubisco located on the gene map. If we can genetically modify this, it can overcome the rubisco catalytic imperfection.
Our solution is to take these rubisco coating strands from the eucalyptus tree and form a recombinant plasmid, which is inserted into E. coli. The recombinant plasmid will then mutate over several generations in direct evolution, and the most efficient mutations in the fixation of CO2 will be selected through intensive screening.
Chloroplast DNA coding for rubisco we modified and inserted into the eucalyptus species. Studies with modified rubisco to enhance food production have shown catalytic improvement of 30 to 45%, and photosynthesis rates increasing as high as 55%.
There are three main selections in our solution. Direct evolution was chosen to ensure an efficient mutation. Biolistics was chosen as the best method of introducing the DNA. And fast-growing eucalyptus trees were chosen because they are just the best CO2 collectors in the world.
Taking this solution can significantly lower atmospheric CO2 if results achieved in food and tobacco are replicated in eucalyptus species and current global eucalyptus species are maintained well. We could reduce approximately 114 million tons of carbon dioxide from the atmosphere each year.
Genetically modifying rubisco may mean the trees grow taller and overshadow other species, leading to ecosystem imbalance. Smaller genetic diversity may also weaken the tree's resilience. People may not want genetically modifying trees growing near them, so appropriate practices are essential.
Modifying the rubisco is biologically feasible, as it has been done before. Similarly, societal issues may arise with these GM plants. However, such plants are managed successfully within the community. While there are issues that may arise with biolistics within the cell, with correct management, it should be fine. And there are high costs associated with establishing this program, especially with plantation rollout.
By incorporating regulated growing zones and also using genetic markers, we can minimize the spread of these genetically modified trees into the wild gene pool. Greater nutrient and water uptake, which also poses as a potential concern, can be omitted through close monitoring in the laboratory environment.
Any questions?
Same sort of question I asked before. It could take some time to grow eucalyptus trees to the point in which they be effective carbon sinks, right?
So we predict probably 15 years to get to that stage, to get to the point where they're absorbing enough carbon to be reducing atmospheric carbon levels.
And how would that work in other climates around the world?
Well, eucalyptus trees are actually planted in lots of places. So there's big plantations in China, in Chile, and Sri Lanka. We could use these plantations, and it would not only benefit the world because they absorb more carbon, but these plantations are also used for commercial wood industries, so they could also be faster growing and thus benefit both the economy there, and also these species are koala feed species, so it's actually good for koalas. There's actually another slide. But it's good for koalas, so these koalas can now eat these, and they grow faster, providing better food source for our critically endangered friends.
So how are you going to convince the governments? I guess this will, definitely you see a lot, there's a lot of benefits, but I feel like in the, especially in Australia, do people want to make change to our,
Yeah, there are many different societal perspectives on GM plants, and GM regulations are different in every single country. So what we're planning to do is be completely transparent about the regulations we're using, how we're going to be testing them in the lab, and the standardization we're using. And also by testing any unknown side effects in the lab before we even consider sending out the GM plants out into the ecosystems and environment.
I don't think that. Also, just one thing to add is that 95% of cotton in Australia is genetically modified, so chances are right now you're wearing genetically modified cotton. So even if some people in the community don't like it, it's quite here already and very present in Australia, and I think that a lot of transparency would help with that.
Can you give me more information about how is it going to benefit koalas?
So the specific eucalyptus species that we've chosen, which is E. clowenziana and E. grandis, are species of eucalyptus which koalas use as their food source, as their shelter, and they basically live and use these eucalyptus trees every single day, which will ultimately benefit them.
So I have to say, I absolutely love all the statistics that you've used to motivate your argument in different aspects. You know, just the numbers you're quoting, really well done. I was also thinking about the koalas, but I wanted to ask you, so eucalyptus is notoriously noted in genetics for being very diverse, right? And I'm wondering, is there any concern about competition from other types of eucalyptus plants that you're aware of?
Well, similar studies of modifying Rubisco have occurred in the US and for different types of trees, and those trees actually grew taller than the ones that were not genetically modified. So we think that these ones are species that already grow to 50 meters tall, so they're already towering over other eucalyptus species. So making them a bit taller wouldn't have as bad an inverse effect as maybe other species would.
What if we could use genetic engineering to stop humanity's most dangerous predator, the deadliest animal on the planet, responsible for the deaths of billions, the mosquito? Mosquitoes aren't just a nuisance, they are the carriers of some of the world's most devastating diseases, including malaria. Malaria is one of the cruelest parasites on Earth, possibly the single biggest killer of humans in history. In 2015 alone, hundreds of millions were infected, and almost 500,000 people died. But a new technology could help us eradicate malaria forever. To do so, we need to engineer a whole animal population. This is not a hypothetical problem. The modified mosquitoes already exist in a lab. The question now is, should we use this technology? Is malaria bad enough to risk it? Malaria is caused by a group of microorganisms known as plasmodia, very strange single-celled parasites that completely rely on mosquitoes to survive. Malaria always begins with an insect bite. In the mosquito's salivary glands, thousands of sporozoites wait until the insect penetrates your skin. Once inside, they head straight for the liver, where they quietly enter large cells, hiding from the immune system. For up to a month, they stay in stealth mode, consuming the cells alive and transforming into their next form, tiny, drop-like merozoites. These merozoites multiply, generating thousands of themselves, and then burst out of the cells, heading into the bloodstream to find their next victims, red blood cells. To stay unnoticed, they wrap themselves in the membranes of the cells they've killed, killing someone from the inside and then using their skin as camouflage. Brutal. They then violently attack red blood cells, multiplying inside them until they burst, repeating the cycle over and over. This process releases pieces of dead cells and toxic waste material, which triggers a powerful immune response, causing flu-like symptoms, high fever, sweats, chills, convulsions, headaches, and sometimes vomiting and diarrhea. If malaria reaches the blood-brain barrier, it can cause coma, neurological damage, or death. When another mosquito bites an infected person, it picks up the parasites, starting the cycle all over again. Mosquitoes are the perfect carriers for human diseases. They've been around for at least 200 million years. There are trillions of them, and a single mosquito can lay up to 300 eggs at a time. They are practically impossible to eradicate and serve as the perfect parasite taxi. The African region carries the majority of malaria cases and fatalities, with the region in 2022 having 94% of all malaria cases and 95% of death. In particular, Nigeria contains 26.8% of all deaths in the world, with an estimated 76% of their population living in high transmission areas.
Our proposed solution is modeled after Project Needlenose in 2015. Project Needlenose involved the release of 750 million genetically modified mosquitoes throughout Florida, using CRISPR-Cas9 technology, which successfully limited the number of malaria cases per thousand. Evidently, our area of focus will be Nigeria, as it contains 26.8% of malaria cases worldwide. Thus, we propose to establish a genetically modified population of mosquitoes of at least 1 billion mosquitoes to be released into Nigeria. This will ensure that the developing mosquito population is an area of high rate transmission. CRISPR delivers a Cas9 nuclease complex which can edit a cell's genome for a synthetic production of RNA that is guided to the required location of the genome. As a result, this can disable or repair existing gene sequencing.
Project Needlenose used the Cas9 technology to edit this gene. The mosquito Down syndrome cell adhesion molecule gene, yes, that is an actual gene, referred to as the AGD-SCAM gene for short. This gene has a complex genome organization, boasting 101 exons that can manipulate their positions to produce 31,000 alternative splice forms. Splice forms are different variations of the AGD-SCAM gene. Each of these forms is used to combat and resist infectious parasites by producing antibodies specifically made to combat them. One of these parasites is the one carrying malaria, Plasmodium falciparum. Cas9 technology will be used to boost the number of AGD-SCAM gene splice forms and their capability in combating malaria, creating a set of female Anopheles mosquitoes immune to the Plasmodium parasite. These mosquitoes will be released into breeding grounds across Nigeria to breed with the local population. As a safeguard, the edited AGD-SCAM gene will also be dominant, ensuring that the edited gene prevails over the recessive original gene during the breeding process, and thus ensuring that the vast majority of the bred mosquito larvae inherit the malaria resistance. However, we accept that this model has several limitations. For a start, the needle-nose model risks the potential of the Plasmodium parasite evolving to override the updated AGD-SCAM gene's new splice forms, which, given the parasite's capability to easily adapt to its new surroundings, is very likely. As such, we present Plan B, which attempts to use CRISPR tech to either reduce the population of or wipe out the female Anopheles mosquito entirely. This plan is based on the work of a biotech company known as Oxitec, which released 5 million genetically engineered friendly male mosquitoes into the Florida Keys in 2021 and successfully reduced mosquito populations in that area by 80 to 95%.
So, what's the secret? Oxitec friendly mosquitoes are programmed with the dominant lethality gene, more accurately known as TTAV. TTAV encodes a protein that blocks the capabilities of other genes that mosquitoes need to develop or survive. Mosquito larvae encoded with this gene will die before maturing. Oxitec raised a large set of mosquito larvae in water containing tetracyanine, which suppresses the effects of the TTAV gene. As such, once an Oxitec mosquito matures, it can breed with the local population, passing on the TTAV gene to its offspring to inherit. Without access to tetracyanine in the wild, said offspring will die. Under the NMI's Plan B, genetically engineered mosquitoes encoded with the TTAV gene will be released into areas of high mosquito populations across Nigeria. Preferably, the NMI does not want to rely on Plan B due to the ethical and practical issues of wiping out the Nigerian Anopheles mosquito population entirely, such as the actions said extermination would have on the ecosystem. So ultimately, thus, the NMI wants to prioritize work on CRISPR editing of the AGD-SCAM gene. Thank you.
Any questions?
Amazing. Cool, where to start? So can I ask, both for Project Needlenose and the Oxitec project, did they monitor the genetics of Anopheles during that time? Like, were there any other changes in those mosquitoes in response to the GM-modified ones?
Okay, so the thing with Needlenose and Oxitec is that they had a very complicated testing system. So the first thing they would do is track the immune system of the mosquito onto computers. So they'd actually run computer simulations of different splice form and exon combinations to see what their effect on mosquitoes would be. The first thing they'd check for is, obviously resistant to the Plasmodium falciparum parasite. And the second thing they would check for is other effects it might have on the rest of the body. Once they found a suitable one, they would then take that to live testing across different mosquitoes. And if that turned out to be A-okay, then they'd test it in the field.
Okay, and then do they go back into the field and sample? Because it looked like it was a fairly decent period of time.
Yeah, exactly.
Did they sort of see any natural variation in the Anopheles in response to these GM ones being released in the wild?
So when they did the testing, they didn't use it over a long period of time. They only tested over like a short period of a few months. So evolution couldn't have impacted our testing results.
Okay. It's interesting though because there's all this data right on species which have really significant differences in their sex chromosomes, right? Like how they're evolving in different ways. So I wonder what that might have on the population.
I mean, like, we accept that it's obviously a bunch of things that could go wrong. It's obviously very new technology, you know, it's only come around for a couple of years. That was one of the reasons why we actually developed Plan B, because Plan A has so many issues with the testing that it might actually just be easier to wipe out the mosquito entirely.
Yeah, that's true. Is there any thought to, just one more question, about like, so it's the female mosquito that bites, right? So is there anything to do with like messing with that, where you still got female carriers, they're just not transmitting the parasite anymore?
So basically, for the females one, we make them infertile, so they can't even breed at all. So basically, within one generation, they're all wiped out.
Yeah, I think it sounds amazing. And I would like to ask, so what are the limitation challenge for using for this initiative? Can you see any issue that can stop this from happening?
Yeah, so ultimately it comes down to our ability to edit this AGG scam gene ultimately, and how effective it will be to sustain in the ecosystem. And ultimately, when done over a long-term period, we will have to have consistent monitoring checks to make sure that there are no other side effects within the mosquito that hinder everyday like quality performance, and that ultimately we will be making sure it's targeted upon the AGG scam gene and its proliferation of gene splice forms to make sure it limits the transmission to specifically humans.
On that note, if it does come down to Plan B, we also have countermeasures in place to make sure that ecological niches that would otherwise have been filled by Anopheles mosquito would be filled. For example, we could find replacement prey for the predators to eat, and we're not too concerned about the pollination side of things because they don't seem to have that big an impact on Nigerian pollination.
Great stuff. Any possibility that this approach could be employed for other mosquito-borne diseases?
Yes, we have actually looked at how there's also other genes such as like the PFAT gene or something that can be edited within mosquitoes, again with concerns to malaria because it's such a big disease, and how the PFAT gene can also be edited through Cas9 technology. Ultimately, so yes, there are other techniques to ultimately prevent the development of malaria and the carry and for mosquitoes to carry it. But ultimately, we are focusing on the Anopheles mosquitoes and the spread of Plasmodium falciparum.
Even within the AGD scam gene itself, there's actually a variety of ways that its exons can be manipulated to focus on diseases that aren't malaria. So for example, dengue fever, you can actually change the exons and program them with the same testing requirements in the AGD scam to make sure that those splice forms create antibodies specifically for dengue fever or otherwise.